Ready.
Excellent. Okay. So does this look about right? About where we left off?
Yes.
Okay. So we're talking about polysaccharides, right? And we're talking about glucose. We left off talking about how you can link alpha glucoses together, and we're going to take a look at what happens with beta. So one of the things I promised on the previous slide was looking at the differences that you get between using alpha glucose versus beta glucose. And so this is going to be alpha glucose. We're going to take a look at the starches. So these are the digestible nutritional types that we talked about before. And so when you link your alpha glucoses together end to end, the 1 carbon to the 4 carbon, you're going to get an alpha 1,4 linkage. Right? Remember, that was the one that links together 2 hexagonal monosaccharides, in this particular case, 2 glucoses. And when you do that, you're going to hook them up end to end. It's going to be basically the 1 to 4 orientation. Now, when you do that, and you basically link these end to end, what you create is a long chain of glucoses. And this long chain of glucoses will actually, as they kind of form, this is just because of the angles of the bonds and just kind of the way they kind of point to 3-dimensional space, will kind of form this sort of a helical kind of a spiral molecule a little bit. And this is actually amylose. So this is actually plant starch. Notice the suffix OSC. So we know it's a carbohydrate because of that suffix. But this is basically your plant starch. Now, another thing that you can do with plant starches, and if you're wondering what these are actually cells like in a potato that are full of starch granules. So these little brown things are like these they're like little pockets, like little balls of just this amylose, all kind of wadded together in a little granule and tossed into this empty cell. And so that's what you're looking at in this micrograph. But one of the things you can also do with plant starch is you can actually form this 1,6 linkage. Now, if you recall, the 1,6 linkage is the one where you form branches, right? So in this particular case, this is your 1 carbon, 2, 3, 4, this is 5, and then this one is 6 right here. This guy's going to be 1, 2, 3, 4, 5, and then this is 6. So in the 1,6 linkage, you're going to take the oxygen associated with the number 1 carbon of the branch, and it's going to link to the 6 carbon. So this guy right here, that bond right there between the oxygen and the number 6 carbon, is this 1,6 glycosidic bond. Now notice it's going to form this side chain. It's going to be branching off laterally to the side chain. You can see this over here in amylopectin. So if you take a look at amylopectin, you can see the main coil, the main chain is basically moving in this direction. And then off of the side, you see this little lateral chain going off into the opposite direction. So it kind of forms a little bit of branch. That right there is a 1,6 glycosidic bond. And this represents amylopectin. And so this is basically, starch. That's basically what it is, like kind of a higher order of plant starch. So if you were to take a look at sort of amylopectin, we actually see that in cooking in the form of fruit pectin. So fruit pectin is this kind of branched starch. That's what it is that you tend to find in fruits. So that's what fruit pectin is. And most of these starches, actually, if you take a look at it, whether it's fruit pectin or if it's some other starch, in culinary practice, they have a common role. They play a common role in the sense that they're thickeners, right? So they kind of tend to thicken things. Instead of being just a runny liquid, they'll kind of thicken it up and give it a little bit more base to it. And so that's kind of what those guys are for. And that's actually what pectin is. So it's basically a type of branched plant starch or fruit starch if it's fruit pectin. Now, generally speaking, plants don't do a lot of branching, and they don't have to, right? Because what they do is, if they lay down these long coils of starch, they can like basically take some of these cells, like these empty cells here, and they can just fill those up with just starch. Not only that, but when you're looking at a plant, and we talked about this last time, right? Remember, a plant can actually take something like a root, like one of these little rootlets, and it can actually sacrifice that root and just basically fill that up with starch. We could do that for several of these different roots. It doesn't really have to worry about it because if it needs more roots, it can just grow more. So plants can pretty much grow their structures whatever they want, however they want. So any given plant can just basically grow out a little rootlet and turn that into this big sort of starch repository. And that's kind of like what you have when you have something like a potato plant, right? So you basically have these tubers underneath the ground, which are roots that have been modified to be storage warehouses, essentially under the ground. And then as a result, they don't have to structure their starches. They don't have to branch it a lot because they can just lay down these individual coils in these large potato structures, and they can make these potato things as big as they want. So there's no problem there. Now, in animals, we can't really do that, right? The plan for our body is already established. It's already set down. So, we can't grow structure beyond our plan, right? So what a plant can do is they can add, branches and leaves however they want. They can add roots through the course of their life if they need to. We can't do that, right? We can't grow another finger or add extra fingers if we feel like that's going to be helpful. Yeah, we have a plan for 4 fingers and a thumb on each hand, and that's it. We don't waver from that. We don't get more. We don't get less. And so the plan is set for us. So we don't have a lot of ability to sort of add structure beyond our plan. Plants have this sort of programmed plasticity to them, and this allows them to be able to adapt to their environment as they wish, as they see fit. But because of that inflexibility that we have as animals, that basically means that we don't have the same kind of capacity for plants to just store starch wherever we want, right? We can't just stick a bunch of starch in our finger because our finger is busy doing other things, and that's true for everything in our body. Now, when you take a look at animals and animal starch, glycogen, in animals, we have the same kind of setup. So we have the kind of coiled branch, the coiled chain of glucoses, right? So that's our main chain. But in animals, it's a little bit different because they have more branches. So like here you can see, here's one branch coming off. Here's another branch coming off here. There's another branch coming off there, and a third branch coming off there. So you can see that there's a lot more branching going on in animals. Matter of fact, if you kind of have your little main chain here, you're going to start to add these branches. Typically, you don't have very long main chains. But what you'll see is that typically the branches that we put on there will start to actually have branches on them. And not only that, but the branches that are on the branches start to have branches. And the branches on the branches start to have branches on the branches. And so pretty soon what you start to see is that you start to get all these branches sort of sticking off everywhere. Anywhere you can put a branch, everywhere you can spin off with this kind of 1/6 linkage, you're going to do it. And so ultimately, you get to the place where you kind of start to have a hard time figuring out where the actual main branch is. You're not quite sure where that main branch was anymore. This whole thing right here is what's referred to as a glycogen granule. It's highly branched, and it doesn't really have like a root main chain. And that's what you're seeing in this micrograph. So these little black dots are actual glycogen granules. I believe this is an image of liver cells. So you tend to sort of store these granules in your liver and your skeletal muscle. And because we don't have a place to store it, right? So that's kind of what we have to do, to basically store it in our liver and then store it in our skeletal muscles. But here's the thing though, what happens if we have more, if we have more capacity, we have like more glucose and we have to store more, if we fill up our liver and we don't have any more liver space to go, and we fill up our skeletal muscle, we have any more skeletal muscle to go. What if we have a bunch more glycogen left over, bunch more carbohydrate left over? What do we do with all the excess? Well, if you fill up your short-term starch reserve, your glycogen in your liver and your skeletal muscles, then what we do is we basically kick it over into fat reserves. So any excess carbohydrate that does not get moved into liver or skeletal muscle begins to get deposited and stored in the form of fat in the fat cells, right? And this is kind of like our strategy, the animal strategy for maintaining our kind of long-term energy reserves in our fats. Notice plants don't have fat. They don't get fat, right? But what they have as their long-term storage is the ability to form things like potatoes, right? Tubers and things like that they can sort of store lots of starch in. So they don't need fat because they have a different type of a long-term strategy. Okay, now let's take a look at and foil this against what it would look like if we took, instead of alpha glucose, we took beta glucose. So what if we actually linked beta glucose together in a long chain? And remember, beta glucose is still glucose, right? And it's still going to be a 1,4 linkage. So that's also true as well. The difference with beta glucose, though, because of the orientation of hydroxyl group above the plane of the ring, what's going to happen is in beta glucose, every other bond is going to be alternating. It's going to be pointing upward instead of downward. That's the big thing. So these little bond areas in between glucose is going to have a slightly different shape to it. Now, what that gives us, because of that subtle change in orientation, is that gives us the ability to stack these glucoses end to end, but it's going to give us a straight chain instead of a coil. As a result, that means we can basically pack these little glucose fibers in tightly to each other and form like a very strong rope-like structure, kind of like this little fiber over here in the micrograph, which is a fiber of cellulose. It looks kind of like a little bit of a rawhide. And that's exactly what cellulose gives us because we can pack it together very tightly. Now, the other thing with cellulose that we also have, remember, is that this cellulose is something that we cannot digest. And we talked about this before, right? We talked about the nutritional starches. We have enzymes for that. And then the cellulose and the non-nutritional ones, the structural ones, the ones we can't digest, we just simply don't have enzymes for that. Now, what I want to do is illustrate for you what I mean by that is to kind of show you exactly what the enzyme for the plant starch does for us versus what that same enzyme would do for us if we were to sort of give it cellulose instead of normal plant starch like amylose. Okay. So normally if you have plant starch, you have an enzyme that breaks it down. So here's my enzyme. And enzymes basically all have a structure associated with them. And it's a three-dimensional pocket, right? So all enzymes have this kind of three-dimensional pocket in them that kind of fits sort of like a hand and glove kind of lock and key sometimes you hear as well. But it's this three-dimensional pocket has a very specific shape to it. And this specific shape is only designed for the substrate that this enzyme works on, right? So in this particular case, if this enzyme is specific for amylose, then it will only bind to amylose. So it only has three-dimensional pockets for amylose. So let's take a look at this guy. So these three-dimensional pockets, these three-dimensional active sites will basically have a nice little three-dimensional pocket that will fit this glucose here, and it'll be happy there, right? And then it'll have like a little bit of a deep depression there to fit that bond angle. Then it'll fit the next glucose. Then it'll have the next bond angle, the next glucose, and so forth. And then everything is happy because everything fits, right? Now at that particular point, then the enzyme will be able to break up the amylose and break it into individual glucoses. Now the problem is we don't have an enzyme to break up cellulose. Matter of fact, the only enzyme we have would be the one for amylose. So if we were to use this enzyme, the same enzyme, let's take a look and see what it would run into. So as it basically forms this three-dimensional pocket, the first thing it's going to form is it has a downward depression to fit that downward angle. But you notice that angle is upward. So there's a non-fit there. This glucose will fit nicely. This downward bond angle will fit nicely. This glucose will fit nicely. But then it has a downward angle, right? It's expecting a downward bond, not an upward bond. So this is also a non-fit. These areas of non-fit is enough to pretty much mean that this enzyme can't do anything with this particular substrate because it doesn't fit. I think we've kind of all experienced this before, haven't we? We've all had keys that slide into a lock, but then when we try to turn it, it goes nowhere. So it's close, but close isn't good enough in this case. It has to be perfect. Everything has to fit. And so basically the reason why we can't digest cellulose is because we don't have the enzymes for it. Now, and we talked about that before, right? Because we talked about, the termites have the enzyme. Excuse me, the termites have bacteria in their guts that have the enzyme that can do that. And so we don't have it, but other organisms do. Okay, so that's the end of carbohydrates. Any questions on carbohydrates before we move into proteins? Okay, everybody's good? Good. So let's move into our proteins. So proteins are really important because pretty much proteins do just about everything in the body. And so when we look at proteins themselves, they have a couple of different functions, right? So they have okay, bear with me 1 second. I'm having like a very strong deja vu moment here. Did we talk about proteins? Okay. Because the deja vu that's hitting me is functions. It seems to me like we talked a little bit about the protein functions. Is that right? That's what I thought. So basically, my notes didn't save from last time, it looks like. Or they only partly did for some reason. Okay, anyway, I'll figure it out. So one of the it kind of yeah, I have no problem going over because this is something that does bear that needs to. We want to make sure we kind of get all this down a little bit, right? So basically, when you're taking a look at proteins, you've got pretty much proteins are doing all of the work in the cell, and so pretty much any process that you're doing is essentially going to be driven by proteins. That's what it's going to be driven by. So for instance, if you take a look at, and this is just a short list, right? So if you take a look at the functions of the proteins, obviously proteins are enzymes, so they're doing catalysis, right? They're driving chemical reactions. That's a big function of proteins. A lot of times we'll talk about defense, right? So your antibodies, these guys are also proteins. Transport, this is both inside the cell and for the organism itself, right? Support, this is both our skeleton and the cell skeleton. Think about it for a second. Your skeleton, right? The tissue that makes up your bone a lot of that, it's calcium and phosphate, certainly, but the real heavy lifting part of your support system, like your skeleton, your cartilage, your connective tissue, is actually wrapped around protein, right? Collagen, for instance, in particular, is a dominant protein in all of your supportive connective tissues, including your bone, your cartilage, and your connective tissue proper, like the stuff that's in your skin and your tendons and your ligaments and things like that. That's all protein. And not only that, but the cell inside of it has a cytoskeleton. So it has like an internal framework that's also made out of protein that gives the cell shape. And we'll learn more about that in the next chapter. Motion, both cellular and organismal. What does that mean? That means if you want to walk down the road, you need proteins to do it. If a cell wants to move from point A to point B, it needs proteins to do it. Regulation. Basically, this comes in the form of protein hormones, like insulin. Insulin is a type of protein hormone. It's a hormone, but it's a regulator of your blood glucose. And then of storage, the biggest one here are the lipoproteins that store cholesterol, which is a lipid. And you've got other types of lipoproteins. These are just some of the functions. Pretty much every function you can think of in the cell or in the body is largely driven by a protein. And of course, how do you build proteins? The polymer, you build it with the monomer. The monomer is the amino acid. And we've already seen this one, right? So every amino acid has an amino group. Got it? Check. Okay. It also has on the other side, right? This is your amino. That's your functional group. On the other side is going to be your carboxylic acid. Got that one. That's the carboxyl group. Check. In the middle is going to be this central carbon, right? The alpha carbon. And attached to it is going to be on one side a single hydrogen. Got it? Check. And then there's going to be an R group, right? Of which you have 20 different choices. And these 20 different choices is what basically sets the amino acids apart. So in every single amino acid, central carbon is the same. The amino group is the same. Carboxyl group is the same. The single hydrogen is the same. The variable group, this is the one that sets you apart, right? So all of this here is all the same for all the amino acids. So then when you take a look at the amino acids, you can break them you can group them according to their temperament, right? It's the R groups that give these amino acids their personality. So for instance, you have major groupings. So you have your non-polars, right? That's basically if you have sort of carbon, hydrogen rich R groups. You have your polar uncharged, typically will have oxygen or nitrogen or both in your R group. Why? Because they're electronegative. Right? We learned about that. Oxygen and nitrogen are electronegative, so they tend to form this polarity, right? This negative-ishness because of that electronegativity. So that's what makes them polar. You also then have charged. Usually it's going to be a negative oxygen or a positive nitrogen, right? That's basically what you're going to have. So this would often be referred to as an acid. This is a base or a basic. Sometimes you'll hear them referred to as acidic or basic. And then you have aromatics, which basically means you have some sort of a ring, usually a benzene ring with the alternating double bonds. Okay, so that's what that means. And then, of course, you have your outliers, the unique ones, proline and cysteine, which kind of has their own little thing. So proline kind of has a non-aromatic ring, so that's kind of special that way. And cysteine has a sulfhydryl group in it. So let's take a look at this. Did you guys see this figure before? Did I show you this? I know I showed this to one of my sections. I don't remember if it was you guys or my Tuesday, Thursday section, because you guys are really close, right? So this is, if you recall, just to kind of refresh your memory, and I think I know where I left off with you guys, just to kind of refresh your memory. Remember, you can group these guys according to chemical personality. So the non-polars moving down the column. They're all true because they're nonpolar. If you take a look at them, they're all nonpolar because they're all these R groups are all rich in carbon hydrogen bonds. Carbon hydrogen, mostly carbon hydrogen, carbon hydrogen, carbon hydrogen, and so forth. Oh, so I just moved. That's lovely. Okay. Okay. Wait. No. Yes. Okay. If you also take a look at this, notice this is mostly carbon hydrogen. Yeah, you got this nitrogen in there, but it's mostly carbon hydrogen, mostly carbon hydrogen, mostly carbon hydrogen, with the exception, this one's a little bit weird because you got this sulfur group in here, but you can see big chunks of carbon hydrogen in there, right? That basically means they're non-polar. For the polar charged, that's going to be this column here. You can see all of them have either an oxygen or a nitrogen in them. So you can see you got your OH there, you got an OH there, you got a nitrogen and an oxygen there, nitrogen and oxygen there. In tyrosine, you have an oxygen there, and then you have a sulfur and cysteine. So these guys are the polar ones because they've got these oxygens and nitrogens in them. And then, of course, you have your charged, where you can see you got a negative charge on oxygen there, positive charge on nitrogen there, negative oxygen, positive nitrogen, and then there you got a positive nitrogen. So that's your charged group. Also, by the way, polar and hydrophilic. So these guys here are hydrophilic. And of course, these guys here are hydrophobic. And then, of course, going across the rows, you group them according to aromatic. These guys are aromatic. You can see they got these big rings on them that have double bonds alternating on them. These do not have any rings on them, so they're non-aromatic. And these guys are kind of neither here nor there. So these guys are kind of like the special outliers that kind of just are their own thing, right? They're their kind of own creature. Okay. Now, I think where we last left off was this slide, I believe, if I remember correctly. We had talked a little bit about the construction of the peptide bond. So here's one amino acid. We're going to link it to another amino acid. And so what we're going to do is the enzyme that's going to come in here is going to break this bond right there. And it's going to break the hydrogens off of the amino group. And so these two hydrogens and this oxygen are going to leave as water. And that's going to leave the carbon to bind to the nitrogen. So the carbon of the acid side of one amino acid to bind to the amino nitrogen of the other. And it's going to form a bond right there. And this is what's referred to as a peptide bond. Now, what I want to do is I want to draw a little bit because the one thing I want to do is we're going to get into the levels of structure. But what I want to do is I want to sort of draw like a tetrapeptide or a pentapeptide, just a generic chain. And we're going to start this off with, first of all, our amino groups. So there's an amino. I'm going to put the nitrogen over here, maybe another nitrogen over there. This is going to be attached to a central carbon. And it's going to have R1 there. We're going to call it R1. It's going to have an acid. And then you're going to have your peptide bond, but it's going to be connected to the nitrogen of the next adjacent amino acid. So we're going to put that one there. Central carbon with a hydrogen. And the next R group, there's number 2. Let's do another one. I'm going to put the central carbon. I'm going to put the hydrogen down here. And you have R3 with your, I'm going to do one more. Okay. So now what I have is a tetrapeptide. Where are the peptide bonds? There's one right here. There's one right here. There's one right there. Now, a couple of things that I want you to notice is that when you're taking a look at kind of a long chain of amino acids like this, you notice there's a repeating unit. There's NCC, 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 NCC. This is a repeating unit that you see. We call this one the backbone. So when you kind of circle that backbone, then what you see is this sort of chain, and then sticking out from it are all these different little groups. Some of these groups are the double bonded oxygens. Some of them are the hydrogens associated with the nitrogens. And then, of course, some further yet are going to be these R groups. So this basically becomes really important to understanding our structure. So let me kick up the slideshow again, and let's start talking about the 4 levels of structure. The first level is what's called primary structure. Oftentimes it's abbreviated as kind of 1 degree. Basically what this is the linear chain of amino acids. Linked together by peptide bonds. So what I actually drew here is primary structure. So that would be the primary structure for this little tetrapeptide. So primary structure, the first level of protein structure is pretty basic. Now then, let's move to the secondary level of structure, oftentimes written as 2 degree. So what happens in the secondary level is basically the backbone hydrogens and oxygens begin to interact and form hydrogen bonds. So let's go back and take a look at this. So notice, nitrogen is electronegative, yes? So what's the charge going to be on nitrogen? So if nitrogen so if it's electronegative, right, it's pulling electrons toward it. So that's going to make nitrogen what? Negative. So if it's pulling electrons toward it, just like with water, right, it's going to pull electrons toward nitrogen. But then what does that mean for the hydrogens? If all the electrons are going toward the nitrogens, in all these cases, all these electrons are going toward the nitrogens. So this guy's going, all those electrons are going toward the nitrogen. What does that mean for the hydrogens that they're attached to? The hydrogen is going to be slightly positive, yes? So you've got these slightly positive-ish hydrogens. Now, what about the oxygens that are sticking off the acids? What are those going to be like? So is oxygen electronegative? So what's the charge going to be on those oxygens? Yeah, those oxygens are come across negative, right? Just like with water, when the oxygen pulled the electrons toward it, those oxygens on those acids are going to be negative. Well, look at what you've got on your backbone. You've got a tetrapeptide held together with peptide bonds, covalent bonds. And you've got all these charges sticking out of it. And so what happens in the secondary level is these charges start to see each other. So like the positives will start to basically see the negatives. So this positive charge will start to see, negative oxygens, and they'll start to fold up on each other. So, so like if you have a peptide like this, and there's like a negative oxygen over here and a positive hydrogen over there, what's going to happen is these guys are going to see each other. They're going to want to bind to each other. And what's going to happen is it's going to bend this backbone so that the positive and the negative are next to each other. And so it folds the backbone up. So when you take a look then at this level, when we're talking about backbone interactions, that's what's happening. The oxygens and the hydrogens on the backbone are starting to bend the backbone around as they form associations with each other, these electrostatic associations. Now, as it turns out, there's two major patterns that you see in secondary structure. One is alpha helix. And basically what this is a coil. The backbone coils like this, and it's held together with these hydrogen bonds between oxygens. The other major pattern that you see is what's called the beta pleated sheet, where it kind of forms like a zigzag pattern like this, where this is held together with hydrogen bonds. So those are two major patterns that you see in secondary structure. And we're going to see some pictures of this here in just a second, so you can kind of get some visual on it. So once your backbone starts to interact with itself and folds itself up into this alpha helix and into this beta pleated sheet, then it goes to the next level of structure, which is tertiary, which is referred to as this third degree. And this is kind of like taking all that alpha and beta sheet and like folding it all up. Like if you were to take a piece of paper and just crumple it up, that's kind of what tertiary structure is like. When you do that, it tends to sort of form this globular protein. So you'll see this kind of like crumpled up, piece with these different types of motifs on it. And it just kind of has all these different little secondary structure pieces to it, some coils, some sheets, and it's just kind of all wadded up in this big globular ball. Now, in some cases, for single enzymes, for single subunit enzymes, this is the final step. So you're done. So if they only have one single polypeptide chain, that means if they're only one single subunit, then you're done. So you act as a lone ranger. It's like an individual sort of a thing. You're done, and you can go and do whatever it is that you do. You're finished here. However, a lot of enzymes are not solo shots. They're part of a team. That brings us to the next level, the final level of protein structure, which is the quaternary structure, the fourth degree. So basically what this is a team of subunits. That are each folded into tertiary structure. A good example of this is hemoglobin. Hemoglobin is what's called a tetramer. Tetra, four things. And it's composed of 4 different subunits, each of which are individually folded in tertiary structure. You have 2 alpha globins and 2 beta globins. An alpha globin by itself is not functional, nor is a beta globin by itself functional. Hemoglobin is only functional if all four of these subunits come together and act as a team. Once they're assembled as a team, then they've acquired their final level of structure, quaternary structure, and then they become the functional hemoglobin molecule. So it's kind of what it looks like. So here you can see your primary structure with your backbone, your R group sticking out, and then of course your negative oxygens and positive hydrogens that then fold on each other, and they basically locate each other. And this is a good example. I love this image because you got the beta pleated sheet. Notice how it's kind of like folded into like these little pleated sheets, but they're held together with the hydrogen bonds. So that's this guy right here. Those are the H bonds. And then the red is oxygen and the white is hydrogen. So you can see you got little hydrogen bond there between oxygen, hydrogen, oxygen, hydrogen, oxygen, hydrogen, oxygen, hydrogen, oxygen, hydrogen. So they're basically hold together with these electrostatic hydrogen bond attractions. The helix, same basic thing, only in this case you form a corkscrew and you hold the elements of the corkscrew together with the hydrogen bonds. So notice you got one there, you got one right there, you got another one right there. That's holding this little ribbon together. There's another one there, another one there, and there's another one there holding that side of the ribbon together, and so forth. So you have these hydrogen bonds between, notice the nitrogen, hydrogen. So there's the nitrogen of the amino group, and you're talking about the hydrogen of the amino group binding to the oxygen of the acid group. And so those are backbone interactions. Now, the important thing, and I don't know that I mentioned it, but I want to go back and write it down just so that I can write it down. Too far back. Is in tertiary structure. What's driving tertiary structure? In tertiary structure, the important thing in tertiary structure is this is where the R groups interact and drive backbone folding. So in secondary, we had the backbone folding causing the folding. In tertiary now, the R groups are starting to interact with each other. And this is where that was important to go over the different types of chemical personalities of each of the R groups, right? Because what we mean by this is if there is a nonpolar next to a nonpolar, then it's going to want to come together. It's going to bend the backbone to do that. If you have a nonpolar next to a polar, it's going to want to get away from it. So it's going to bend the backbone in order to do that. So it goes through all these contortions so that these interactions between R groups, polar with polar, nonpolar with nonpolar, nonpolars run away from polars, things like that, can basically form their associations with each other. And as they do that, they're bending this backbone and they're continuing to fold everything up. And that's how tertiary becomes this globular mess. It's like a really bad game of Twister. And so that's what's happening in tertiary structure. The R groups interact. And as a result, what you're going to get is a big globular protein. So you notice you have a series of different little things here. So you have like, here's some sheet here that breaks into a helix, and then some more sheet, another helix, and then a loop, and then helix, a little bit of a loop. Here's a little mini sheet, little helix, mini sheet, helix, little baby helix, small run. And then you kind of got this big sheet-like structure here. And that would be one subunit. And then in quaternary structure, you can see that each one of these little globularly folded subunits forms a member of the four-person team, and that would be quaternary structure. So this one would be secondary, this is tertiary, and this is quaternary. Okay, so let's take a look at what we call motifs, right? So motifs are basically commonly occurring secondary structural patterns. And not only do they occur often, but they're associated with function. So oftentimes, if we see like a secondary structure pattern, and then we studied a particular protein that has a particular function with that. Let's say, here's a good example. Let's say we take a DNA binding protein. And there's part of this protein that's designed to bind to DNA. Well, when we study this protein, we notice that there is a particular secondary structure motif. Like maybe it's a helix loop helix, right? Something like that. And that this is the area that binds to the actual DNA and make contact with the DNA. So what we've noticed then is, as we study other DNA binding proteins, we notice that when in their DNA binding domain, they have a similar helix loop helix pattern where they make contact with DNA. And we've seen this over and over again so many times to where we call this now a motif. And not only that, but if we take a look at protein X, like if we discover protein X, like some new protein that we've never even studied before, and we notice a motif, that we can basically say that, okay, if you see a helix loop helix in here, then that means protein X must bind DNA, because the helix loop helix is a DNA binding motif. That's what it uses to bind to DNA. And that's kind of what motif analysis of proteins has given us. We can determine the function of proteins by taking a look at what sorts of common motifs they have in them. And that'll kind of tell us some things about what they do. Now, domains are a little bit bigger. So these are like motifs, but they're bigger. And they tend to be functional areas within a protein itself. So, but domains are just larger areas with, you know, multiple different types of motifs in them. So here's a good example of what we mean by domains versus a motif. So a common motif, like for instance, is beta, alpha, beta, right? So here's one beta sheet, here's an alpha helix, and here's a beta. So you have the two alpha 3, right? So the 2 sheets with a helix followed by 3 sheets. So that's a common occurring pattern that we see that's associated with some sort of a function. And then we can see helix loop helix or helix turn helix. And so this is your alpha helix, here's your turn, and then here's an alpha helix. And this we associate with some sort of function as well. Now, when we're taking a look at domains, generally speaking, a domain is a little bit bigger. A domain might have some recognizable motifs in it, but it doesn't necessarily have to. So domains can be made of anything. But generally speaking, domains are like modules of function. That's what they are. So if you take a look at this, I'm going to circle the domain. So here's one domain right there. Here's domain number 1. And then we have domain number 2 right here. And then we have domain number 3. And this could be domains like within one globular subunit, right? So this is like one globular subunit. So let's take a look, maybe that's part of a quatermeric a team structure, like a tetramer or something like that, right? So it kind of comes together as sort of a member of a team. Well, what might these domains do? Well, here's just an example of how these domains would work. So let's imagine that you've got this tetramer and it's got, like, 4 different types of subunits in it. And in each one of these subunits is going to be some certain functions, right? There's a series of functions that each single subunit has to have. So for instance, domain number 1 may be designated as what we refer to as the catalytic domain. This is the one that does like whatever chemistry you have to do, whatever chemical reaction you do. So this is like the catalyst section, right? This is the one whose job it is to make whatever chemical reaction that you're responsible for go forward. In domain number 2, this one might be the regulatory domain. So this is how the subunit takes instructions. So you got outside instructions telling this particular enzyme what to do, how to do it, when to do it, how much to do. All those instructions will be taken in by this regulatory domain. And typically those instructions come in the form of proteins themselves. So this is an area that might interact with other proteins as those proteins are trying to communicate what the set instructions are, like how much this particular enzyme is supposed to make. And the third domain, because it's a member of a team, could be what we refer to as an oligomerization domain. What does that mean? So this is basically, to oligomerize basically means you're kind of coming together as a unit. So this is basically where the subunit would actually hold hands with its partners in crime, right? So if it's going to be part of a tetramer, a team of 4, then it has to tether itself physically often to its teammates. And so this domain might be responsible for reaching out and actually tethering itself to its teammates so that they can form a foursome. And so here you can see you have 3 very distinct functions that this particular subunit has to be able to carry out. It has to do its chemistry, catalytic domain. It has to take instructions, regulatory domain. And it has to actually physically bind and become part of a team, the oligomerization domain, or the tetrameric domain. There's a couple of different terms you can use for that. And so this is what domains would do. And then within a domain, we might identify motifs of function. And that's kind of how that flows. It's kind of like a nested hierarchy. Okay. Any questions on general protein stuff before we move into chaperones? How are you guys doing, temperature check wise? No news is good news. So far, so good. Gotta love emojis. So let's because we're talking about folding, so let's take a look here and see what this all comes down to. Remember, there's a little bit of history here that I wanted to sort of sketch out. And that's the way we used to think this worked, and the way we actually have discovered how it works now. A good example of maintaining your objectivity, always being open to modifying your ideas so that you can grow and learn better and understand things better. But we used to believe that as soon as you made the linear chain of proteins, that folding was for the most part spontaneous. That is to say that essentially it kind of happened on its own, right? And there's a good reason for that. I mean, it makes a lot of sense, right? Because think about it. I mean, when you're forming hydrogen bonds, you don't have to do anything for it, do you? I mean, water naturally sticks to itself. It just kind of happens, doesn't it? And so when you take a look at the R groups, for instance, those are also spontaneous, aren't they? I mean, if you put a nonpolar next to a polar, what's going to happen? So if you put a polar molecule next to a nonpolar molecule, what's going to happen?
They repel each other?
Yeah. Do you have to do anything for that repulsion to happen?
No.
It's automatic, right?
Yes.
So if you think about it, since a lot of our R groups are mostly polar or nonpolar, it makes a lot of sense, right? I mean, it's reasonable, isn't it? That if they're next to each other on a backbone, they would just automatically do their thing, right? They would run away from each other. That seems reasonable. So we used to think that this was all automatic, right? That basically folding was all automatic and everything folded up by itself. Not only that, but we used to think that every now and then that it would kind of fold in correctly, right? And when it folded incorrectly, we had to have proteins come in and fix it, right? So we used to think that these used to come in, chaperones used to come in to fix the rare unfolded protein. Like, for instance, we used to think, okay, all this should happen spontaneously because the whole polar non-polar interaction is spontaneous. And it should, and normally like 80-90% of the time it's fine. It gets it right, right? But then maybe 10% of the time, 20% maybe at most, it just kind of flubs up a little bit. And the idea is, it's like, well, my guess is if we just kind of straighten it out a bit and we kind of let it do it again, it's probably not going to make the same mistake twice, right? So the second time around, it'll actually fold properly. And so we discovered these chaperones as proteins. We used to think that a chaperone was largely just a fixer, right? So it's like, okay, for the unusual unfolded protein, then we have the chaperones that just come in and kind of tweak them a little bit, fix them a little bit, and then they'll fold nicely. However, what we noticed was that chaperones are a bigger deal. And how did we notice that? We created deficiencies in chaperones. Basically, we deleted genetically chaperone genes. And we asked ourselves, what happens? Well, what we found out was that instead of just like seeing, maybe a slight increase in the number of unfolded proteins because nobody was fixing them, like maybe a 10% or a 20% increase, what we saw was a catastrophic fail in protein folding. What does that tell us? It suggests that chaperones are much more involved. They're not just fixers. they're required to actually get these things folded properly. And it makes a lot of sense, doesn't it? Think about it. I mean, if you guys go to prom, like back in the high school days, and you have all these chaperones all over the place, I mean, what does a chaperone do? I mean, do the chaperones at these events, I mean, are they just like, do they just sort of like show up and then disappear for most of the night? Is that how those things went? If you can remember back that far, if you went to prom?
Yeah, that's how it feels almost.
Yeah, right. I mean, in some cases, yes, but guess what? Was there no supervision at all there?
Oh no, that was always there.
Somebody was watching, right? The principal, the teachers, somebody was always watching. There, when you kind of stripped it all back, and how did you know this? Well, you knew this because if you wanted to do something squirrely, guess who was basically breathing down your neck, right? Making sure you towed the line, right? So that's nobody wants to have a complete drunken brawl fest of a prom on their watch, right? That's a principal who pretty much is going to lose their job the next day, right? Nobody wants that one. So the chaperones at like a dance are like there all the time. It's like they're watching all the time. They're like deeply involved in the whole process of it, right? And this is kind of the way the chaperones are for proteins. They're not just fixers. They don't just sort of stop and go and just sort of drop you off and take off. They're like there all the time, and they're actually the ones facilitating the events. Oftentimes they're the ones who are emceeing the events. They're the ones who are making everything move along and progress nicely. So they're deeply involved in the entire event, and that's what we found with these chaperone proteins. And what we found when we saw some chaperone deficiencies were some real diseases like cystic fibrosis. Cystic fibrosis is actually it's a failure of a chloride channel, and in this particular case, actually, this chloride channel is a protein. It's a transport protein for chloride ion, and it fails to fold properly. So the protein is misfolded, right? So it's otherwise normal, but it's a misfolding thing. Why? Not because the protein, right? Here's the thing. This is important, right, for this one. So it has the correct amino acid sequence. So if our original idea was that these things spontaneously fold if we have the sequence in place, then this shouldn't have a problem, but it did. Why? Because it's not the sequence itself that allows it to fold, but it's the chaperone who's actually in there doing the folding. So you can imagine the chaperone as almost like a potter, like on a potting wheel, basically with its hands on the clay, molding and forming the clay. And the chaperone doesn't have to do much. Just like with a potting wheel, you don't have to do a lot, right, to get the pot to shape up. All you have to do is kind of put your finger right in the middle of the ball, and it automatically starts to form kind of the cup shape. So you didn't do very much. So it looks like the clay is kind of magically doing its own thing, but it's actually not. It's your finger that's actually helping it to do what it needs to do. And the same thing is also true here. The chaperone looks like it does very little, but what it does do is really critical in order to get this sequence to fold properly. And that's kind of what it does. So here's your chaperone protein. Typically, you'll take a misfolded protein or an unfolded protein. It'll enter into one chamber of the chaperone, and usually there's like dual chambers, so you can like work on multiple proteins at the same time. And then, generally speaking, you'll burn some ATP, so it's an energetic process that'll put like a little cap on it. And then the inside of this will kind of work and massage this misfolded protein until it's able to kind of work it into its correctly folded shape. And then once it does that, it'll uncap itself and it'll release the correctly folded protein. And then, of course, what can it do? It can basically go back and grab another misfolded protein and kind of get it folded up again. And so these chaperones are really critical in making sure that these proteins are getting folded appropriately. Now, the reason why we put so much premium on folded proteins is because for proteins, three-dimensional shape is everything. It's super, super critical, right? Think of a protein's reliance on three-dimensional shape as kind of like a key's reliance on three-dimensional shape to be able to fit a lock. So if your key doesn't have the exact three-dimensional shape that it needs to have, it's not going to be functional in your lock. The same thing is true for the protein, right? If it doesn't have that exact three-dimensional shape, which it gets as it folds up, then it's not going to be functional at all. And so oftentimes when you're folding the protein up, you're creating function as you're creating that shape of the protein. Now, what happens if the protein loses that fold up three-dimensional shape? What happens if it unravels, right? If it misfolds? Well, if it misfolds, it's going to lose its structure, its three-dimensional structure, and its function, right? Because in this case, three-dimensionality equals function. When it does that, we call it denaturation. Basically, the unfolding of a protein. So there's a couple of things, a couple of parameters that will affect a protein's function because it affects its folding. And that's what we have here. So the three things are temperature, pH, and salt concentrations. So what we're going to do is we're going to take extremes of these, and we're going to kill the protein, right? So if you do these in drastic enough measures, you will kill the protein. Now, I want to mention this though, because you don't have to kill the protein to make it non-functional, right? All you have to do is just slight adjustments to these, and you can make the protein non-functional. We saw that, didn't we, at the end of the buffer talk? Remember we took a look at the buffering range, and we said that beyond a pH of 6, this enzyme is no longer active because it's out of the necessary active pH range that it needs in order to be active. Well, notice we didn't kill that enzyme, right? We didn't unravel it completely. We just made its circumstances suboptimal so that it's not really functional anymore, but it's still alive. I mean, it's still, it still has its structure. It just doesn't have the circumstances or the environment that it needs in order to be functional the way it needs to be. And so in smaller adjustments, these same parameters can affect the productivity of an enzyme. These are just the extremes that we're looking at here. Okay, so let's take a look at extreme conditions. pH. So extreme pHs can denature proteins. An example is make cheese with lemon juice. What's the pH of lemon juice? Do you guys remember? Two, yeah, right. So it's really acidic. Now, have any of you guys ever made cheese before? Sometimes I get somebody who's made cheese before. It's actually a fairly easy process. Basically, you take milk, and not like the healthy stuff. You want to take like real milk. I mean, typically good cheese is made from raw milk, right? So you want to have milk with the milk sugars and the milk fats in it, and lots of protein in it, right? So you want to use either whole milk or raw milk typically. And generally what you do is you put some sort of a curdler in there. So normally milk is made with an enzyme called rennin. which is actually from sheep gut. But sometimes some individuals, they want more like a vegan type of a cheese, so they use, instead of rennet, because it comes from an animal source, they use lemon juice. And so because of the pH of lemon juice, what it does, it denatures the proteins in the milk. Those proteins coagulate, they tangle up as they unfold. They cross-link with each other. They get tangled up like a really bad string of Christmas lights, right? They get hopelessly tangled up. And then what it starts to form is these insoluble globs of protein and milk fats and milk sugars that we call curds, right? So when little Miss Muffet sat on her tuffet eating her curds and whey, what was she eating? Anybody know what she was eating? Curds and whey? For you nursery rhyme fans? You guys are like, I have no clue what she was eating. Whatever curds and whey is. So the curds are the individual solids after it curdles. And the whey is the liquid portion that's left behind. So curds and whey is basically cottage cheese. Now, when you get it to curdle, to get the milk to curdle, and we've had milk curdle before, probably. Like if we're cooking and we have too much heat or something like that, we oftentimes can make our milk curdle, just like when we're cooking. It's usually an undesirable thing. But in this particular case, all you do is you kind of strain the curds away from the whey with cheesecloth. Actually, cheesecloth, that's what it's used for. And then you compress it, get all the liquid out of it, maybe put a brine wash on, a salt wash on it, and you let it age, and that's your cheese. But the way you did it was by denaturing the proteins, first of all, with lemon juice. That's extreme pH. In temperature, extreme temps can denature. So the example here is cook an egg. So let's go ahead and do that. Let's crack an egg on a frying pan, a good hot one. What does the egg white look like in an egg when you first crack it in there?
Clear.
Yeah, it's clear. And then as it cooks, then what does it look like? Is it still clear after you cook it?
No.
Yeah, it's opaque. You're right, Cadence. It's opaque and white, right? So what happened there? Well, as it turns out, in egg white, as we mentioned before in lab, remember in the macromolecules lab, we use albumin. There's a lot of albumin in egg white. And so that's a protein. It's a folded protein. And so when you have that extreme temperature, it basically overcomes all those hydrogen and bonds, and then it unfolds and unravels a protein, and then they get cross-linked together, just like they did with the cheese situation, and they get tangled up hopelessly, and then you can no longer refold them anymore, and so they're just kind of hopelessly a tangled mass, and that becomes that opaque white egg white. And so that's basically what happens when you're cooking. You're basically denaturing proteins. Okay, and then if you have really strong, salt concentrations, you can denature a protein as well, but those typically are a little harder to come by, mostly. So usually what you see in denaturing is pH and temperature. Those are the big two. So one of the things I always like to kind of run by you, because when we're talking about temperature, right, and when we're talking about cooking, you cook an egg, and you denature the protein. I always like to lay this question out there for you guys to just think about for a second, because eggs are interesting, aren't they? You can cook an egg solid all the way through, but how many of you guys like eggs, like over easy, where the yolk is runny?
Yeah, that's very nice.
So a lot of folks will like that, right?
Hmm.
Do you know anybody who's actually like swallowed eggs, like drank eggs, like raw?
Yeah, some gym rats.
Yeah, some right. Some people do that. But here's an interesting thing. If you think about it, if we can eat portions of the egg raw, which is basically what the runny yolk is, or if some individuals can just swallow the egg raw without cooking it, then why are we cooking the egg?
That's a good question.
Actually, if you think about it, why are we cooking anything? I mean, take a look at nature. We're mammals, right? Take a look at all the rest of the mammals out there. Do any of the other mammals, carnivores in particular, do they cook their meat?
No.
No, they eat it raw, don't they?
Hmm.
So why are we the only creatures on the planet that cooks our food? Why are we cooking? What does it do for us?
Taste, I guess.
Let's taste, but that's largely psychological, right?
Mm, true.
I mean, if you omit that part of it, I mean, obviously you're right. Food is a very psychological thing. But if we just ignore that for a second, just from a biological perspective,
Mhm.
is there any real need to cook anything? I mean, think about it, right? Do vegetables have proteins in them?
No. Really?
Oh, they do.
They do? Oh.
Hmm. They have proteins,
Oh yeah. Yeah, yeah, yeah.
right? How about nuts?
Yes, that's right.
Nuts have protein in it.
Yes, they do.
Do we have to cook nuts?
No.
Not really. Do we have to cook vegetables? A lot of us prefer our vegetables raw.
True.
So how come we cook food? Because after all, think about it for a second. The albumin that's uncooked and it's clear is folded up. That's the only difference between the albumin that's cooked. So just from that perspective, structurally, I mean, is there any real benefit to cooking? And yes, Cadence, you hit the nail right on the head. There's only one reason why we cook anything. It's not the albumin that we're trying to cook and denature. It's not the meat that we're trying to cook and denature. The only reason why we cook anything is because the proteins we're trying to denature are the bacteria that live on our food. That's the only reason why we cook. If it weren't for bacteria, we would be able to eat all of our food completely raw. Which is kind of interesting because normally we think of cooking not as a safety measure, but we think of cooking as like, oh, well, we have to cook this in order for it to like, it's like not worth eating or we can't eat this if it's not cooked. It's like, no, you can eat it just fine. As long as there's no pathogens on it, you can eat it just fine because all it is just protein. But of course, not going to be eating raw food anytime soon. Why? Because I don't want to have to go and have my stomach pumped. Okay. So that's denaturing. Any questions on proteins before we move into the next molecule?
Are the chaperones proteins job only to untangle stuff?
Who's the chaperones?
Yeah.
Yes, that is their thing. That's what they focus on, is unfolded protein problems.
And
So
they are other proteins, right?
Huh?
And they are also proteins.
They are themselves also proteins, right? So it's, I mean, like it's a kind of a little bit of a circular sort of a thing, isn't it? So what happens if your chaperone has a problem folding? So it's like the folder is not folding, so they can't fold if it's unfolding, right? So it's like it becomes very circular, but that's kind of part of, and actually that circularity, which is kind of, and you kind of hit on something that's really interesting actually from kind of an evolutionary developmental sort of a thing, is because it gets to one of those chicken or egg kind of moments in evolution,
Sure.
which, to be quite honest with you, evolution has no answers for. And they struggle with that part because it's like, oh, wait a minute. So in order to fold a protein, you have to actually fold it with a protein, but the protein itself has to be folded. So who folded the folder? Right? So it's like, if you don't have a folder full of protein, then how did it get folded in the first place? So you can see it runs into this sort of chicken egg thing. There's several of those moments in evolution, and nobody really has any good ideas about how that exactly worked. That's kind of one of those, head scratching moments that we run into every now and then
Got it.
that are fascinating, but we're not quite sure exactly how that went down. But those are like interesting little side notes. Okay. Any other questions before we move into nucleic acids? Okay, off we go then. Nucleic acids, right? So the polymer here is the actual nucleic acid. The monomer for a nucleic acid is called a nucleotide. Now, nucleotide, very similar to an amino acid, is kind of has modules to its construction, right? So the modules in a nucleotide are composed of three different things. Just like an amino acid has three different things, the nucleotides have three different things. One is the sugar, right? The carbohydrate at its stop. That's kind of its base. And then to this base carbohydrate is attached a phosphate, which is one of the reasons why we talked about phosphates as a functional group early in the chapter. And then on the other side is a nitrogenous base. And there's different types of nitrogenous bases there. So you have these three different pieces of every single nucleotide. Now, depending on which nucleic acid you are, you will have a different sugar. For instance, DNA has a deoxyribose. Remember we talked about that? These are the 5-carbon sugars that we saw in the carbohydrate section. Deoxyribose is the foundation for DNA. Ribose is the foundation for RNA. We'll talk more about the structure of DNA and RNA, especially when we get to the replication chapter toward the end of the semester. Now, in the bases, these nitrogenous bases, what we have here are basically 4 different types of bases, and they're grouped into 2 major families. The first family is what's called the purines, and they compose 2 bases called adenine and guanine, A and G. Okay, those are the 2 purines. These are basically double ring structures. Now, the other family of bases is what's called the pyrimidines. So the pyrimidines are single ring structures. And they compose thymine, T, cytosine, C, and then an RNA only is uracil. So these are all of our nitrogenous bases that we run into. Now, what happens then is when we have our nucleotide monomers, we connect these guys together with dehydration synthesis, and they're connected by a bond called the phosphodiester bond. So there's the name of the bond that connects nucleic acids, right? So we had glycosidic bonds connecting carbohydrates. We had peptide bonds connecting amino acids. we have phosphodiester bonds connecting nucleotides. And so this is what a nucleotide looks like. So here you can see your ribose sugar. And the two different versions of this is all centered around this 2' position. If that 2' position has an OH group on it, then that is ribose. And that is the stock sugar for RNA. If, however, it terminates in a hydrogen, that is deoxyribose. And that is the stock sugar for DNA. Now, to the 5 carbon is going to be attached a phosphate group. So you can see this is a typical phosphate group, right? It's bound to these 4 oxygens. One of those is double bound. And then on the number 1 carbon on the other side is going to be this nitrogenous base. So you can see here, this particular nitrogenous base is the purine type, right? So you have these 2 different rings, 2 fused rings together. Matter of fact, this 2 fused ring system I use to help me remember what the purines look like, because I think purine, I think Purina, right? The Purina double dog dish. So remember Purina used to put out those little connected little double dog dishes? And this is kind of like your little double dog dish, your Purina double dog dish. So that's your purine. And that's your basic nucleotide. Now, this kind of shows you structurally what the purines look like. So you can see the double dog dish look here, these 2 fused rings here. That's typical of the purines. This is your adenine, where you can see a big amino group sticking out there. On the guanine, you can see an amino group, but you can also see a carbonyl group sticking out there as well. So those are your double dog dishes, A and G. The pyrimidines, which are the single ring structures, all of them, then we'll have different structures. So if you can see this big electronegative oxygen sticking out there, you can see the amino group sticking out there. On the thymine, you can see these oxygens sticking out there, and you can see a big old methyl group hanging out there. It's the only one with a methyl group out there. That's the reason, by the way, why they have the methyl group on our list of functional groups that we talked about earlier in the chapter. And then the uracil, you can see, looks very similar to thymine, only it doesn't have a methyl group. It just has a hydrogen instead, but you can still see you got your oxygens in the same basic positions. So generally speaking, when you take a look at DNA versus RNA, DNA will have thymines in it, but in RNA, it'll replace the thymines with uracil. And that's the reason why it's only in RNA. So basically, when you take a look at the construction of a nucleic acid, you'll see that it also forms a backbone. As you're connecting these things together, you'll see you got your phosphate right here that's connected to your 5 carbon, your 5' carbon. And it's connected with what's called an ester bond. So that's another functional group in organic chemistry that you don't have to lose sleep over. But an ester bond is basically a situation where you have an oxygen bound to a carbon bound to another oxygen. And so this is what's referred to as an ester. Okay, that's a totally different functional group. There's a whole swath of chemistry associated with that that organic chemists get into. But because this is a carbon bound to an oxygen that's part of a phosphate, that's the reason why they call it a phosphoester. So this is a phosphoester. And then what happens is the other oxygen is bound to the 3' carbon of it, the adjacent nucleotide. So that's the other phosphoester bond. So that's why they call it the phosphodiester bond. And so you'll see that when you're connecting these together, it forms this repeating unit of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So you have this long backbone chain, backbone, where you have these nitrogenous bases sticking out at the number 1 positions. The other thing also to understand is if you take a look down here at the bottom, notice what's sticking out on the 3' end is this hydroxyl group. Why is that important? This is important for dehydration synthesis. Why? Because what do you need in order to add the next nucleotide in line by dehydration synthesis? You need the components of water, right? Remember when we looked at the dehydration mechanism, we said that one of them have to have an OH on it and the other one has to have an H on it. So you have the components of water involved so that you can do your dehydration synthesis. Well, this OH on the 3' end is the OH for dehydration. And the H to go with this comes from the new nucleotide. So this is the reason why this OH is important, because this represents what we refer to as the growing end. So you add your next nucleotide to this 3' end. So generally speaking, in any stretch of DNA or any kind of a nucleic acid, you typically will have a 5' phosphate on one end, and on the other end, you'll have a 3' OH. And this is the direction of growth. It grows in the 5' to 3' direction. And of course, you have your bases here, like A, C, G, T, A, T, G, G, A. And whatever you want, whatever the sequence is. Okay, so let's go ahead and compare DNA versus RNA. So DNA has some differences to it. So first of all, it's double-stranded. That basically means it's got 2 pieces to it. It uses deoxyribose instead of ribose. It uses thymine instead of uracil. Those are all differences between RNA. RNA is single-stranded, not double. It uses ribose, not deoxyribose, and it uses uracil instead of thymine. So notice, because it's a double helix, you notice that basically what you have is kind of 2 halves of a ladder. So here's one backbone with my bases sticking out of it. And here's my other partner backbone, kind of like the rungs of a ladder. And the other thing we know about DNA is the bases actually zipper up to each other with hydrogen bonds. So what we found out was that every cytosine has the capacity to form 3 hydrogen bonds with every guanine. And every adenine has the capacity to form 2 hydrogen bonds with every thymine. This is the reason why on one side, where you see a guanine, it's always paired up with its complement on the other side, cytosine. If you have a thymine on one side, it's paired up with an adenine on the other side, and so forth. So if I were to write a sequence like AGG, ATTA, then what would go on the opposite side of this first A?
A T.
A T, and it would be held together with 2 hydrogen bonds. How about with the G? Who goes with that?
C.
Be C, and it would have 3 hydrogen bonds, and the next one would be, that's a G, so there would be a C again, right, with 3 hydrogen bonds. A goes with T, right?
T.
The T goes with a
A.
A, yep, with 2 hydrogen bonds. This other T goes with an A, and this last A goes with a T. So you have your 2 strands. It's like a ladder. It's like a twisted rope ladder, actually. And all you have to do is give it a bit of a twist, and then you're going to get your double helix like this. That's your twisted rope ladder of a DNA molecule. RNA, being single-stranded, doesn't have to worry about complementation, doesn't have to worry about doing complements, because it's just single-stranded. But notice it's also got this sugar phosphate, sugar phosphate, sugar phosphate backbone. So what do these guys do? right? So what is their function? Well, these are actually informational molecules. These are the sequence for proteins, right? The common question that always comes up is like, okay, we just got done with proteins. We selected a bunch of amino acids in a series, but how do you know which amino acid of the 20 to pick at any given position? That information is encoded in DNA. DNA is the information to make everything in the cell. So both DNA and RNA are informational molecules. And all they are is a sequence of bases, a sequence of A, C, G, T, complemented with each other by hydrogen bonds. So your DNA is actually held together with these weak hydrogen bonds. And so this is kind of what it looks like, your twisted rope ladder. So DNA makes sense, right? It's the complete instructions to make everything in your cell. But what exactly is RNA? So RNA is similar, only that DNA is the master blueprint. RNA is simply a working copy of a part of the DNA. So for instance, RNA will be basically a copy of the information needed to make a specific protein. Just one, just whatever that one protein is, just a specific protein. And so that'll be what RNA is. Whereas the DNA will be the entire set, all proteins in your body, all proteins in your cell, everything. Remember, in your DNA is all the information necessary to build you from scratch, from zero. In that process, you're going to have to use working copies of it, and that's the RNA. So they're very important in terms of information transmission. We'll talk about how they work very intricately together in the last chapter when we talk about transcription translation, because RNA is basically implementing the information, the plan, the design that DNA has. And then ultimately it will transmit that information that will lead to the selection of the amino acids in individual proteins. So there are other groups, however, than just RNA and DNA, other nucleotides, one of which is this guy right here. This is ATP, right? This is our energy currency. This is our like rechargeable battery. This is what we're trying to make when we do metabolism. So if your cell has energy that it requires to do some sort of cellular process, it needs ATP to do that. It's like a rechargeable battery. Just like if you want to use an app on your phone, you have to have a charged battery to give you energy to do that. This is the charged battery for your cell. If you don't have ATP, you're not going to have energy to do what you need to do as a cell. There are also others that are involved in metabolism. These are electron carriers that are carrying electrons in these sort of redox reactions in metabolism that allows you to be able to make more ATP. So you can see that even these nucleotides, NAD plus and FAD plus, are designed to allow you to make more ATP, which is your rechargeable battery. The more ATP you have, the more energy you have, the more you're going to be able to do. So that's a good thing. But these are all nucleotides, different types of nucleotides. So that brings us to our last group of the four, the lipids. But you know what? This is actually perfect timing because we've also reached our witching hour. And this is actually a perfect place to begin when we reconvene, not on Monday, because Monday is going to be our exam, but next Wednesday. That make sense?
Yes.
Okay. Any questions?
So we said DNA is kind of the blueprints to the building, and RNA it's kind of the blueprints to the kitchen of the building.
Yeah, right. So like if you're building a big building, the master blueprint has everything, the foundation, the plumbing, right? Everything. But the guys who are just working on the kitchen, they just have a copy of what they need to know for the kitchen, right? They don't care about the foundation and all that sort of stuff. So they just have a very limited copy of just the project they're working on.
Okay.
That,
And can you repeat what were those two helpers of to create ATP?
NAD plus and FAD plus. So when you guys do the energetics, that's part of metabolism. So the cellular respiration component of it, that's where NAD plus and FAD plus are involved. They're involved in that cellular respiration pathway, which is a big, long pathway to basically be able to make lots and lots of ATP.
Got it. All right. Thank you. Thank you.
Okay. So that is it. You guys are free to go.
Thank you. Have a good day.
You too.